Okay, before we get into the real work for today, I want to remind you of some of the important ideas having to do with the sequencing worksheet rather. Okay, now those are due today, but I know that people are having a lot of issues with it. So I'd like to kind of reiterate the important things for this. Okay, so for example, when we're given a DNA sequence, we're given only one strand, and unless specified otherwise, it reads 5' to 3'. Okay, so when we're going to do either sequencing or PCR reactions, we have to design primers appropriately according to the important rule that the 3' hydroxyl of the primer is going to point into or across the region that we're trying to synthesize, either by chain termination for sequencing or by simple primer extension for PCR. Okay, so when you've got a single nucleotide sequence like this, the first thing you need to do is to envision or even write out the other strand, which is what's shown in my little diagram here. Okay, so we've got our separated strands, and we know from this what primers we need. We need one that's going to point from the 5' end in with its hydroxyl across the insert, and again we want one at the other end with its OH directed across the region of interest. Now looking at these sequences, we can see that the left primer needs to bind to the strand that we don't see and will actually equal in sequence what's present on the upper strand. Okay, the only way that can work is for this 5' to OH primer binding thing can match, which is to say be equal to, the upper strand that we're given and is going to bind to the template, which is the one whose sequence is not shown. Okay, the other primer, of course, needs to bind the other end of the insert. And of course, the insert is running, where the sequence we're given is running 5' to 3' with the 3' at the right. So we can see that the primer that's going to have its hydroxyl pointing in the correct direction is going to read 5' to 3' in the other direction and will be, in fact, complementary to the upper strand and use that strand as a template. Important things to keep straight. The easiest way to do this is just to write it out for yourself and convince yourself. Okay, so what this looks like for our hemoglobin example, I'm going to skip this one, this is written down. All right, is what this looks like. I simply created the other strand. I drew what the template, what the other non-visible strand was going to be. Now you don't have to write the whole thing, but just enough to let you know what's going on. That the forward primer, which is on the left, is going to bind to the strand that's complementary to the one given, and it equals the strand that you are given, which makes sense. So when this primer gets extended, it's going to use the lower strand, the invisible one, as a template, and will create, therefore, a complement to the template. So the sequence that will be created is actually equal to the upper strand, the 5' to 3' that you're given. So for that, with that left primer, the first 3 nucleotides of the template are ACA, for example, and then we're going to complement those, and we're going to get TGT as the first 3 nucleotides of the newly synthesized molecule. Now, it's important to remember that the primer themselves are not labeled, right? They're just synthesized, and we throw them in there, and we ask them to bind their templates, and then we add the enzymes and the nucleotides to then extend those templates. So the primers are not labeled because they weren't synthesized, right? So these are 20 nucleotide primers, which is more typical. The one on your assignment's only 10 for convenience only, right? So we're going to see what the first 10 nucleotides are here by saying after this prime end, 3' end, we're going to be seeing the complement of that because it's being used as a template, and we can assign the colors appropriately. Now at the other end, you've got a similar problem, except kind of opposite, because your primer is running 5' to 3'. So it's actually the complement of the bold-faced sequence in your handout, right? So we're not given the actual primer sequence. We're given the location where the primer will act, but it's not itself necessarily the primer sequence. So we can write the primer sequence by complementing what's given and putting in the correct polarity, which is to say running 5' to 3' towards the left across the insert. Right, so in that case, we're going to be using that strand, the upper strand, as a template, copying it. So the first three nucleotides will be the complement of what you're shown, and that will be T, G, A, G, G, because we're going to be extending that 3' end right to left across the insert or the sequence, right? So just keep your strandedness in mind, and of course, once you do that, you can assign the colors. Okay, now it's not that bad, but if you just don't do anything to the sequence, it's just really hard to see what's happening. Okay, so remind you again, the primers are oligos. They're unlabeled. They bind the template, right? And then they create the new strand. They're not synthesized, so they don't have colors, right? When you start synthesizing, that's when you start getting the colors, because the colors come from the labeled dideoxy NTPs, which are used in the sequencing reaction. Okay, reiterating, we're doing sequencing, so we've got a reverse primer, and that has to have its OH pointing into the region to be copied. So just pay attention to complementarity and what's the template and what's going to be the product, and everything should be fine. And just put in the nucleotides and put in the color as is suggested in the question. It's not that bad. Any questions? Okay, so we're moving on to talking about plasmids more and wondering why we would even go to the trouble of making these things. So there's a lot of different reasons why you want to use plasmids. General cloning for sequencing mutagenesis and stuff. But very often people have interest in, for example, producing a protein that might be of interest, either in E. coli, yeast, or mammalian cells, whatever. The ultimate goal of the plasmid might be to reduce expression of a target gene in mammalian cells, for example. Engineering genome, understanding promoters. There's a lot of different reasons why you would want to put a plasmid together for an experimental goal. And the important idea here is that we need to choose a plasmid vector which is going to be helpful to us or provide things that are not present in your standard minimal plasmids like we've been talking about. But first I want to remind you what we were doing last time. Okay, here's our general typical plasmid. Now, all plasmids have to have origin of replication for E. coli. Needs to have an antibiotic resistance gene for E. coli. And then it needs to have restriction sites in the form of a multiple cloning site where the molecule can be opened up for the purpose of inserting a gene. Now, this molecule, as shown, also has a promoter and another selectable marker, and those can be very helpful, but they're not essential features for absolutely all plasmids. So today we're going to talk about just a few examples of how plasmid vectors that you buy are modified in such a way to further the goals you might have for that particular experiment. So we summarize this: origin and replication, antibiotic resistance, multiple cloning site, promoter, etc. So those are general features of plasmids. So we're going to go for a few of the bells and whistles, which doesn't even begin to touch the surface of the kinds of different kinds of plasmids that are available. Right. We must have origin, antibiotic resistance, multiple cloning. Okay, remind you where we were. We have a double-stranded plasmid that we're going to introduce into the E. coli cell by transformation, and we're going to make many more copies of that purified plasmid for whatever purpose we might have. So that's going to be the first step for almost all these manipulations. But the question is what features this plasmid vector has to have, which are going to further our ultimate goals of, for example, producing or purifying a mammalian protein. Okay, so we need to have restriction enzymes, we need cloning, etc. Here's a picture of a multiple cloning site, also called polylinker. And it does have a whole bunch of different restriction sites, so you have a choice of what particular enzyme you might choose to open up the particular that particular molecule for cloning. And different ones will work differently for different inserts, depending on what sequences are already there and what else you're planning to do with it. Okay, the purpose of the ampicillin resistance gene is to produce a protein that degrades ampicillin, to protect the cell from ampicillin in the medium, to kill ampicillin-sensitive cells. A and B are true, A, B, and C are true. Okay, so the answer is D, which you might not have realized. Okay, so it doesn't really degrade ampicillin. It opens up the molecule so that it no longer interacts with its target. So that basically inactivates the ampicillin. So that A is certainly true. To protect the cell from AMP in the medium is also true, because if a cell contains the ampicillin resistance gene, it will be transcribed and translated in the E. coli cell by E. coli RNA polymerase and ribosomes. It will go to the cell surface, where if it encounters ampicillin, it will open up the molecule, it's a beta-lactamase, and therefore render it inactive. So the cell is essentially protected from the ampicillin by virtue of the fact that it has this new protein, which has been encoded by the AMP gene on the plasmid. So the answer is D. A and B are true. Okay, so we're going to get to the meat of the matter here. What is the purpose of making a particular plasmid? And how would you choose a vector which is going to help you with that purpose? Okay, so there are many different choices, but we're going to just focus on the idea of producing a protein today. You want to produce a prokaryotic or eukaryotic protein in E. coli, or else, in which case you're just sticking with E. coli. You might, however, want to produce a mammalian protein in mammalian cells. So you're still going to be starting with E. coli in this case, but you know in your mind that you're going to be putting that molecule into mammalian cells. And so you would choose a vector which is going to be suitable for mammalian cells ultimately, even though you're going to do your cut and paste using E. coli. Similarly, you might produce protein in other non-mammalian cells, such as plants or yeast or Drosophila or something. So importantly, it's good to have the plasmid vector to have features, sequences, which will allow expression and detection of the protein as part of the vector to facilitate the later steps in the reaction, which are beyond the cloning steps. Okay, so thinking ahead for things that we want to do, if we want to produce and detect either prokaryotic or eukaryotic proteins in E. coli, we need to have a promoter. Now, the prokaryotic gene that you clone may have its own promoter, but people often choose to substitute the whatever promoter is in the vector because they can be stronger and more well-behaved than the natural promoter of whatever gene it is that you're cloning. If you're going to clone a eukaryotic cDNA, you definitely need a promoter because the cDNA does not have a promoter of its own. It's simply a copy of mRNA sequence, so it doesn't have any sequences needed for expression or even termination. So you certainly need a promoter of some sort. Okay, the next thing to think about is what you're planning to do to detect the protein, because you want to know that your clone is successful lots of times, and that how are you going to tell that? Well, an antibody is the best way to tell if you're producing your correct protein using a Western blot, but many proteins don't have good antibodies available for them, and you don't want to spend a whole lot of time and money creating your own antibody. So the easiest way around this unknown antibody situation is to add protein tags to the end of or the beginning or the end of the protein that you're going to try to produce. So tag is extra amino acids or extra almost whole protein things that are appended to your protein to make fusion proteins where the antibodies are available for the tag. So you can detect your protein by adding a few amino acids of a tag to your protein and use the antibodies that are specific for that particular protein. Similarly, if you want to purify the protein, you can do that, but it's a lot easier to fuse it to a tag that binds to an affinity matrix of some kind. And we'll see an example of that in a second. All right. So if you're going to be expressing a eukaryotic protein in E. coli, we want an E. coli protein for sure. If we think we want to express your protein in mammalian cells, you're not going to use your E. coli promoter. You're going to have a mammalian one. It's important to realize that eukaryotic promoters don't work in E. coli. So if you're going to produce your protein in E. coli, it has to be a prokaryotic one. Okay, so sort of envisioning, I've showed this slide before, what's happening. Your blue box refers to your cloned, in this case, cDNA, for example. The rightward arrow indicates the direction that transcription is going to occur across that insert in order to produce the mRNA, which will ultimately produce our protein for us. The purple thing is the promoter. All right, so the promoter is going to bind RNA polymerase, and that will enable the transcription to occur so that we can produce our protein. Now, it's sometimes very helpful to have a regulated promoter because some protein products are toxic or have deleterious effects on the prokaryotic cell. So you want to have a way to turn on or off the production of your RNA and protein. And there are a lot of good prokaryotic promoters where it's understood how to turn them on and off so you can regulate the degree of expression or total lack of expression from the gene of interest. We'll give examples later. Okay, possible popular promoters. The LAC promoter is probably the most popular because it's been around for so long. This is the promoter for the LAC operon, and its regulation is understood and can be manipulated. Another popular one is T7, which is from a bacteriophage, and a hybrid between T7 and the LAC promoter. Okay, if you want to produce a protein in a eukaryotic cell, you need a promoter that works in eukaryotic cells. Okay, now eukaryotic promoters tend to be large, and they're not really very well suited to be placed as part of a cloning vector. So for mammalian cell lines, very often viral promoters are used, most popular being CMV, which is cytomegalovirus. The reason being that virus promoters are relatively small. There must be constraints on the size of promoters in viral genomes, and these are well understood and work in a lot of cell types. Now, they're not all other regular promoters are also used, but the viral ones are very popular. Right, so example, here's CMV, cytomegalovirus, and we've got an illustration of the kinds of organisms which might be the eventual goal or the eventual home of the plasmid that you're making. Yeast, plants, Drosophila, mouse, and human. So if you're going to be taking your molecule and putting it into any of those organisms, you want to make sure you've got a promoter that works in the appropriate organism, and that conveniently the vector would have that on it already, so you don't have to do anything. So CMV is often used. It's small. SV40 is another one, and that's also popular. Okay, again, essential features. We now have to think about the promoter in the cases where we're going to go into a eukaryotic cell and what that would look like. Okay. So how would you know that your protein has been being produced? And this is a little bit more complicated. You need to detect it somehow. And of course, the easiest way is to use specific antibodies along with Western blotting. Good antibodies might be hard to come by or not exist, and this is where your tag is going to be helpful. So the idea is here we'll have cloning vectors where the extra amino acids for the tag are encoded in the plasmid itself and then can be constructed in such a way that what you create is a fusion protein, including your protein of interest, with these extra amino acids kind of stuck on one end or the other, usually either N-terminus or the C-terminus. And that will enable you to use antibodies directed against the tag, and that will then detect the protein that you're interested in. It's a very cute and convenient trick. All right, so it's important to remember what's happening when you have transcription and then translation across a stretch of DNA. All right, the RNA is going to get produced, and then the ribosomes are going to hop on and use the genetic code to produce the protein. So what's going to happen in these situations, you're going to have an AUG somewhere, and that sooner or later you might encounter a stop codon. So what you have to do is get rid of the stop codon to make sure that translation reads through, starts at an AUG, and then doesn't stop until it gets to the end of either the tag or your protein. So this does involve a little bit of manipulation of the sequences at the junction between your tag and your protein. Right, so the idea is we're going to add some extra amino acids, either a few or a lot, and these are the tags, and we're going to use those to visualize our protein later. And this can take some manipulation of a sequence, but that's not hard. Okay, so examples of tags, and these vary widely. The ones that are more or less usually used for Western blots are the small ones where the tag is only a small number of amino acids. The flag tag, for example, the HA tag, the MYC tag, are most commonly used for the purpose of detection by Western, either in prokaryotic or more likely eukaryotic cells. So those are all very small, but they might be put in there in multiple copies just to improve your sensitivity. Okay, these can also be used for purification purposes. Now, more often for purification purposes, GST and the His tag are used because they have good affinity purification matrices available. Now, GST is a large, well, it's not large, it's only 30 kilodaltons or something, but that's a big thing to be sticking on the end of your protein, whereas the regular small tags are only a few amino acids. Okay, so the most familiar example is the GFP tag, which is green fluorescent protein. And this vector shows an example of how you would put your protein to make a fusion protein between GFP and your protein of interest. Okay, so this is intended for mammalian cells, as you can tell from the CMV promoter, which is cytomegalovirus. And then we've got a bunch of restriction sites, which will enable us to clone our gene in front of the GFP gene, which is already included in the vector. So we're going to make a fusion protein. Okay, so you're going to place the DNA in the multiple cloning site, and that's going to make a fusion protein, which will enable us to see at least the location of our protein in the cell. All right, so what this looks like here, we've got our ORF, which is our protein coding region of interest, and you can see that it's been put up there so it completely abuts the GFP sequence. And that's shown in this sequence here. Here's a GFP tag, and here's a bunch of restriction sites that you can use to place your gene of interest, or the ORF, in front of GFP. And you know the reading frame, so you just have to make sure you don't get any stop codons in the wrong place. And so you create a fusion protein, which in this instance will start at the AUG of your protein and run all the way through and include the entire GFP protein. Okay, so here's an example of GFP fluorescent fish. Okay, GFP will fluoresce and look green when it's excited by the correct wavelength of light, but there are variants of the GFP which fluoresce at different colors, and so that's why we have some green fish, some pink fish, and some yellow fish. There are manipulations or variants of GFP that have all those different colors. Now, one incredibly useful thing about GFP is, for example, if you have a protein that you think it might be a nuclear protein, for example, and you make a GFP fusion to it and put it in a mammalian cell, all you have to do is look at the cell and see if the GFP is in the nucleus or not. You don't need any fancy genetic manipulations to do that. Just slap the molecule together and put it in the cell and see what fluoresces, among other uses. Okay, so those fish glow, and we will see more of GFP on a different day. So we're going to talk about a couple of these tags, which are used for different purposes. I'm going to go explain a little bit about GST, which is glutathione S-transferase, as a mechanism for purifying proteins. His tag is also used for this purpose. And then we'll talk about a smaller tag, which is the HA tag, which is more often used just for detection by Western blotting, although occasionally for immune precipitation. Okay, so here's our GST, which is a large protein, and here's our HA, where is it, HA here, which is a few amino acids. Okay, so the reason for using GST is there's a very common and easy to use affinity tag. GST is glutathione S-transferase, and it binds glutathione. So here's glutathione, which is stuck to agarose beads, which can be poured into a column and serves as an affinity matrix. So if GST sees the glutathione, it binds to it in a very tight and easily done way, and therefore binds it. Now, if GST is in the form of a fusion protein, you're going to have the GST part binding, but the rest of the protein is coming along for the ride. So this is a very effective and very easy way to purify proteins. Okay, so what's shown at the bottom is actually a purification of 3 different GST fusion proteins. And you can see that they're different sizes because part of the protein is GST and part of the protein is the protein of interest. But to see how effective of a purification this is, in the left gel, for example, we've got molecular weight markers. And then the first lane after that is the cell lysate. These are E. coli cells. So that's the total proteins that are present in the E. coli cell at the time the GST fusion protein was being made. So you can't see the GST fusion, you can just see a whole bunch of protein. The next lane is the flow through, so that's what the proteins look like that actually did not stick to the column. So the lysate was poured over the column, and the material was washed through, and those are the proteins that did not stick, which is of course most of them, because most of them don't stick to glutathione. Then we've got wash, 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 and finally the next three lanes, E1, E2, are elution, where the column with the bound protein is flushed with an extra of glutathione, which competes for the GST with binding to the matrix. So that basically flushes the protein off the column, again because the GST has been released from the glutathione. And then the samples run, a little bit of it is run on this SDS polyacrylamide gel and then stain. So you can see there's a tremendous purification here. In the original cells, we could barely make out whether GST was there or not. But here you end up with a gel lane, which is almost totally purified, probably 90% purified, of that protein of interest. And then you have two more examples of the same idea, but different proteins. It's a very powerful technique. So the stain gel, it's not a Western, that's an actual stain gel. And it does illustrate how effective of a purification this is. Okay, so in this particular vector, the restriction sites which follow the HA tag are used for cloning your gene of interest, again in such a way that you avoid stop codons, so that when translation starts at the AUG, it does the HA tags, and then it moves along into your protein and makes a fusion protein, starting with the HA and going all the way to your protein of interest stop codon. And in case you're not sure, there's an extra stop codon here in case yours doesn't work for some reason. Okay, so a vector with this has the CMV promoter again. It's going to go into mammalian cells and a bunch of restriction sites where you could stick your protein coding region of interest so that it will be fused, if you pay attention to the reading frame, to the HA tag, which is encoded by the vector. And that will enable you to detect your protein based on its HA tag for which the antibody is available. So these are usually used for Western blots more than anything, but also for immune precipitation. Okay, and the good antibodies are available, and they are used either at the N-terminus or the C-terminus of the protein. It doesn't really matter which in most cases. Okay, so here's our Western blot. Okay, this is a little complicated, but not fatal. All right, so we're going to use Western blotting to detect the proteins of interest using antibodies, in this case to the tag rather than to the protein itself. But the tag is now part of the protein, so that's considered very fair. So the steps in this, first we're going to make an extract of cells that are expected to be producing the protein. This could be E. coli or a mammalian cell, depending on what's of interest. Then the proteins are run on an SDS gel, and then transferred to a filter membrane. Okay, you can't use antibodies to probe an SDS gel because it's gooey and will break. Instead, the proteins are transferred to a membrane filter, which is easily handled. Okay, once you've got your transfer to the membrane filter, it's then probed with first a complicated series of molecules. First, the primary antibody, and that's the one that you purchase to bind to the tag. Then there's a secondary antibody, which will bind to the primary antibody. Now, the secondary antibody is coupled to a detection system, which will ultimately allow you to see the effects of light that was emitted by the product of the HRP. All right, we'll get there in just a sec. Okay, so here's what this looks like again. Here's a stained bands, and then we have the Western blot where we've probed with antibody to the protein of interest. And you can see that it's very easy to see and actually invisible in the actual stain bands from the total cell. Okay, this isn't the best way to show transfer. The better slide is next. But this reminds you what SDS electrophoresis is like. You've got SDS, the gel is between plastic plates, and then the proteins are run up and down the gel, and then they are displayed along the gel according to their size. Now, display is probably a bad word because they're not visible, but they are present in the gel and separated according to their size. So in terms of transfer, once you run your gel, what you want to do is get the proteins out of the gel onto the membrane. And that is done electrophoretically. So here we have our gel, which is green. And we're going to place the membrane on top of the gel, and the membrane is shown blue even though they're white. And then we're going to squish them together with filter papers or sponges with a bunch of buffer and turn on the current. Only this time the current is not running top to bottom of the gel. The current is running from the gel in the direction of the membrane. So what's happening is that all the proteins that are sitting there in the gel are going to get transferred into the membrane simply by electrophoresis. So they're going to electrophorese themselves out of the gel and into the membrane. Okay, so once we have our proteins transferred to the membrane, it's essentially an imprint of what the gel looked like originally, where everybody's still in the same place as if it were bands. So now we have to probe it using antibodies. Okay, so the primary antibodies are directed against the protein we are trying to see, but it's actually directed against the tag that's part of the protein that we're trying to see. Okay, these aren't labeled, but they will bind. Okay, and these will be monoclonal. Okay, so the next thing is our secondary antibody, and they are used to detect the first one. So the secondary antibodies are actually the most crucial part of this, probably. Okay, they're directed against the primary. So if our primary antibodies are monoclonal, that means it was probably raised in mouse. So what we need is a secondary antibody that will detect the mouse antibodies. So in this example, we will have a goat anti-mouse, where mouse IgG was used to immunize goats, and then the goats created antibodies to the mouse IgG. So what that means is that the secondary antibody will detect or bind to any antibody that was made in mouse, for example, our monoclonal. Now, to make things more complicated, these secondary antibodies are themselves tagged with something which can be detected later. In this case, it's going to be HRP, horseradish peroxidase, which has an enzymatic activity which can be detected either by chemiluminescence or by in some cases, in colors. Okay, so the eventual thing that this looks like, and there's another picture later, we have the membrane blot. That's where our proteins are displayed. The triangle indicates the target protein, which is the one that we're trying to detect. Now, the first antibody will bind to the target protein. So that would be our primary antibody, which in this case is detected against HA. Right, but that primary antibody is not easily detectable by itself. Instead, we have a secondary antibody, which is directed against the primary antibody. So that makes the next part of this little sandwich. Okay, now the secondary antibody has been manipulated so that it's covalently linked to HRP, horseradish peroxidase, which has an easily detectable enzymatic activity. So then when we've done all this, we feed it the substrate for the enzyme, and then which is HRP, and that is detected either by colorimetric or chemiluminescent assay. The chemiluminescent is by far more popular, and what that means is that the product of the reaction emits light and is essentially detected by an imager which is sensitive to the visible light that's emitted. Okay, so protein on the membrane, primary antibody binds the protein, secondary antibody binds the primary. The secondary is coupled to HRP, which can be detected by light emitted by the product of the HRP reaction. And then we can see the locations essentially by an imaging film, essentially like taking a picture of it with film. The emitted light is observed, and this appears as a band on the gel, just as if we'd stained it. Okay, another picture of the same thing. And here's another picture of a Western blot to remind you. but this one was authentically done with an HA tagged fusion protein. Lane A has the same cells but with no tagged protein, and B is a control containing the tagged protein. And this is a blot that was probed with the anti-HA antibody, and that was detected with an HRP chemiluminescent fusion protein system. Yay. Okay, everybody have a nice Mardi Gras holiday. Yay.